So we now move into the translation component of this chapter. And translation is the process by which messenger RNA is used as a template upon which to assemble the correct amino acids in the correct order. And this diagram here shows a list of the 20 amino acids that are found in human proteins and their three-letter abbreviation over here on the right-hand side. So we may utilize these abbreviations. An amino acid is made, would you believe, of an amino group and a carboxylic acid group, as well as an R group, which is the variable between these different amino acids. This diagram shows all the particular R groups, and if you look at any one particular amino acid, let's take leucine here. Here is the amino group, here is the carboxylic acid group, and you can see that's the same on all the other amino acids. And the bit in the yellow, that is the R group, and that varies between the different amino acids, and that gives the different amino acids different properties. We can combine these different amino acids into groups showing different properties. So here are aliphatic amino acids, here are aromatic ones, acidic, basic, hydroxyl, so forth, and here's the only cyclic one, which is proline. During the process of translation, we join amino acids together, and here is amino acid number 1, and here is the carboxylic acid group here. Here's amino acid number 2, and here's the amino group. And with any organic reaction, we take this hydroxyl group, this hydrogen, join them together to make H2O, and if we make a new bond between these two amino acids, and this is called a peptide bond. In a similar way to DNA having a 5' end and a 3' end, a chain of amino acids has an amino end and a carboxylic acid end, which is out here off the screen. And so we can say they have a polarity to them, one amino end, one carboxylic acid end. And you can see on this diagram there are multiple amino acids, all with these different side chains, except these two are of course the same, and different side chains give different properties to this particular polypeptide. So what's going to bring all these amino acids together? Well, there's another player in the role of translation, and they are called ribosomes. And ribosomes are a group of proteins that are bringing amino acids together in the right order. They're the link between the messenger RNA and the amino acid. Ribosomes are made of two halves. There is a small ribosomal subunit, of this diagram on the left, and a large ribosomal subunit, this diagram on the right. And when you join them together, here's the small, here's the large, makes an entire ribosome. So let's see what ribosomes are made of. And this diagram shows, here's a large subunit, we'll call this the large subunit, and this one down here is the small subunit. And when we join them together, this is the intact ribosome here. But let's look at the components. We find that a large subunit consists of some ribosomal RNA. Two components actually, this size and this size. There's a large and a small bit of ribosomal RNA, plus a whole bunch of ribosomal proteins gives you this unit here. Similarly, another piece of ribosomal RNA, this one 16 units in weight, plus some more proteins gives you the small subunit of the ribosome. Linking the messenger RNA to the amino acid is this molecule here. This is transfer RNA, and it has an amino acid joined on one end, that would be this end here, and it has a special sequence of 3 RNA bases there, and that's called an anticodon. You can draw properly. And this anticodon binds to this here, which would be the codon, and that of course is on the messenger RNA. And so here is this molecule, this transfer RNA molecule. The one end is the amino acid, the other end is the messenger RNA, and it is making sure that the correct codon is binding to the correct amino acid here. This transfer RNA is about 80 nucleotides of RNA long. It is a single-stranded piece of RNA that is folded up on itself into this three stem loop structure. We start translation with an initiation complex, and this is the messenger RNA molecule containing the code, the transfer RNA, which is bringing the first amino acid, and the small subunit of the ribosome. And they form a diagram like this. Here is the small subunit, here is the transfer RNA, and in this particular case here is the amino acid in here. And so when all of these three components come together, then the large subunit will join on top. There's the small subunit, large subunit. This is the initiation process for prokaryotes. Up here. Prokaryotes, in other words, bacteria. For eukaryotes, it is slightly different. In eukaryotes, because the messenger RNA has a cap at one end, the cap is the binding site for the small subunit and the transfer RNA and the first amino acid. These three components bind together and at the cap, and they scan and down until they come to the first AUG. So this scans towards the right, and it comes to the first AUG, and of course you should know that AUG is a start codon there. Once this happens, then the large subunit can bind in on top, and you'll find that the whole ribosome then encompasses the first amino acid, transfer RNA, and the messenger RNA altogether. This ribosome has a number of locations in it. There is something called the P site, and next to it is something called the A site. And the first amino acid always goes into the P site. If you remember this, this is the genetic code, and we mentioned just now AUG as the start codon. So if we look here, here's A, and here's U, which means we are in this box here, and here is G, which means that this bottom amino acid is the amino acid coded for by AUG. And so this is methionine. It's also the start codon, which means that every polypeptide starts with the methionine amino acid. Here's the diagram showing all the various components. This is the DNA down the bottom. Here's the messenger RNA coming out through the nuclear membrane. Here are the small subunit and large subunits of the ribosomes, and this is the transfer RNA with an amino acid, and the transfer RNA goes into the P site. The next transfer RNA comes into the A site. And so here is the start codon, this AUG, and therefore this first amino acid is going to be methionine. The second codon, which is GUG here, is joined by this anticodon right in there. And so you can see we have two amino acids next to each other. Both of these here are inside the P and the A sites. They're right in here. One amino acid, second amino acid coming out of there. Once you've got those two amino acids joining together, you'll get a peptide bond made between the two amino acids with water released. So how do you remember what's going where? Ribosomes, they hold two transfer RNAs at a time, one in the P site and one in the A site. How do you remember what happens at each site? Amino acids arrive at the A site, and the polypeptide is pinned to the P site. There are large subunits and small subunits of ribosomes, and they vary in size depending on whether you are a prokaryotic or a eukaryotic organism. A prokaryotic has a 50S large subunit and a 30S small subunit. Eukaryotes are 60 and 40 respectively, and S is just a unit of weight. And ribosomes are made from ribosomal RNA as well as ribosomal proteins. So once we've assembled everything in line, we now need to keep joining all these amino acids together. And here's our first two amino acids joined together, as we saw in one of the previous slides. Once that first amino acid is joined to the second one by a peptide bond, this transfer RNA is now free, and it can go away. It detaches from the ribosome and from the messenger RNA and disappears. Remember, all this is taking place within a ribosome. Once that first transfer RNA is free, the ribosome now moves such that it goes down 3 bases. And so this is now the P site, and this is now the A site. And so the polypeptide is pinned to the P site, and amino acids arrive at the A site. Once these two are close to each other, they are joined again by another peptide bond, and the system repeats itself. This first free transfer RNA will now disappear, and the next transfer RNA will move into the P site. This system continues until you end up with a series of amino acids all linked together. So here's some text about what actually happens here. The new word here is peptidyl transferase. This is an enzyme. Specifically, it's a ribozyme, which, if you remember, it means it's a piece of ribosomal RNA that acts as an enzyme. And peptidyl transferase is the enzyme that makes that peptide bond between the amino acids. Those free transfer RNAs that are released from the ribosome, they are recharged. In other words, they return to another enzyme called amino-acyl transfer RNA synthetase, and they are recharged with the correct amino acid so that they can go back and repeat their job. Here is a diagram showing what's going on. Large subunit, small subunit. This is the P site and the A site. Some texts also include an E site that stands for exit. It really just means that's where things leave from. This is the first amino acid attached here. Second amino acid comes in. Peptide bond is joined between them. These are other factors called elongation factors. The whole ribosome then moves down 3 bases, such that this second transfer RNA is now in the P site, and the A site is now free to allow a second transfer RNA to move in to then join peptide bond between those two as well. This is the amino-acyl transfer RNA synthetase enzyme. This is the transfer RNA, and here is the new amino acid. And this enzyme, its job is to make sure the correct anticodon on this transfer RNA is joined to the correct amino acid over here, and it makes this bond. So you now have a charged transfer RNA. So how do you stop translation? We have 3 ways of stopping translation, and they're given right here. These are all stop codons: UGA, UAG, and UAA. There's no transfer RNA that binds to these codons, but there is a termination factor, and this termination factor binds to the A site over these codons, causing the ribosome to fall apart and release the protein it's already made. Remember the genetic code? Again, we've got those 3 stop codons. Here they are. Here's 1, here's 2, here's 3. And if you look, they are U, AA, UAG, and UGA. Once we have made a polypeptide, that's not the end of the story. The polypeptides need to form a 3-dimensional shape so that they can perform a function. And so these shapes are sometimes assisted by other proteins called chaperones.